Without knowing anything about DNA sequence, it's often possible to determine genotype just by looking at phenotype. In this photo, all three brothers have blue eyes, and since blue eye color is a recessive trait, you can guess that both parents also have blue eyes, and they do. Family pedigrees can also indicate a lot about the inheritance pattern of the trait. In a pedigree, females are the circles and males are the squares. Each row of symbols in the pedigree represents a generation of the family. Traits are indicated by different kinds of shading or other markers. In this pedigree, eye color is indicated by the color of the symbol. We can designate genotype by the letter B. Big B equals brown or the dominant trait, and little b equals blue or the recessive trait. At the top of the pedigree is a blue-eyed mother and a brown-eyed father. Mom has blue eyes, so her genotype has to be little b, as do all the family members with blue eyes. Dad has brown eyes, so his genotype can be big B or big B little b. Since Mom can only pass a little b to her kids, and since they all have brown eyes, they all have to be big B little b. You can actually infer all the rest of the genotypes from these that we see here. Since we know the genotype of the mom next to the arrow, and we know that one of her kids is little b, her husband must be big B little b, and their kids must have at least one big B allele. You can determine inheritance patterns for Mendelian traits by going through a series of questions. The first question to ask is, are only males affected, and are all sons of an affected father also affected? Such an inheritance pattern would be a Y-linked inheritance. All males receive their Y chromosome from their father, so no daughters will be affected, but every son will be. Unfortunately for men, if their dad had hairy ears, they probably will too. The next question you can ask is, are only males affected, and is the trait passed from females to males? In this case, this is an example of X-linked inheritance. Males receive an X chromosome from their mother and a Y chromosome from their dad. Since males only have one X chromosome, any defects or alterations on that chromosome will show up as an X-linked trait. And color blindness in men is one of the most common X-linked traits. The next question you can ask is, do any offspring with the trait have both parents who are unaffected or without the trait? If the answer is yes, that would be an example of autosomal recessive inheritance. In an autosomal recessive condition, like the one shown here, affected individuals must have two parents who are carriers, and therefore be unaffected. In this pedigree of leptin deficiency, which is a protein that regulates eating behavior and energy balance, those who are half-shaded are designated as documented carriers, and individuals A, B, and D were affected with leptin deficiency and severe morbid obesity. In fact, 8 members of this family were presumed to have had leptin deficiency due to severe obesity and early death, which is designated as a line through the symbol, during childhood, including a 6-year-old girl, person E, who died before the initiation of treatment. It had been known that mouse models with this defect could be treated with recombinant leptin and their normal body weight restored. Individuals A, B, and D are shown in the photos on the right, before and after leptin treatment. It's amazing what success these individuals experienced with just simply replacing the missing protein. The next question you can ask is, do all affected offspring, regardless of gender, have affected parents? In this case, this is an example of autosomal dominant trait. Dominant traits are found in every generation. Dominant traits are also often found in genes that encode structural or developmental proteins, as in the case of brachydactyly or shortened fingers or toes. Another example of an autosomal dominant trait is Huntington's disease. This is a devastating progressive neurodegenerative disorder with a late onset. Prior to the discovery of the genetic defect driving the disease, individuals didn't know whether their offspring would develop the disease or not. The discovery of the Huntington's disease gene enabled parents to make informed decisions about future children. This disease has no treatment and is particularly challenging because it actually due to an expansion of a CAG repeat sequence in the gene, which would be very difficult, if not impossible, to target with gene-based therapy. Finally, you might ask the question, is there only one affected person in the youngest generation in the pedigree? This would be an example of a de novo mutation. In this pedigree, although one of the grandparents is designated as obese, he has a BMI of only 30, while the proband, designated by the arrow, has a BMI of 62.5 at age 13. This extreme phenotype is likely the result of a new mutation not found in either parent. We've walked through several examples of recessive and dominant traits. As shown in this table, 2 copies of a gene have to be disturbed to see a phenotypic change in the case of autosomal recessive traits, while only 1 copy of the gene can be disrupted for a dominant trait to emerge. While these rules of thumb for identifying inheritance patterns are very helpful, not all traits are inherited in a Mendelian or single gene fashion. Codominant patterns of inheritance exhibit an intermediate phenotype for the heterozygote. It's not an on or off trait as in Mendelian inheritance. And unfortunately, most traits in the human genome exhibit this more complex pattern. Thus, mapping DNA sequence variation to a complex trait like obesity, for example, is exceptionally hard. Remember that the DNA sequence variation can be in genes and promoters and regulatory elements and non-coding sequence. And so that's one level of complexity. And then, of course, there's the complexity of the environmental factors that may be influencing the phenotype of interest. But with new statistical tools and new approaches for identifying these interacting factors, there is hope that we can do a personalized assessment and intervention for individuals in the future.